I'm going to show you how to test for protein, lipids, starch, and finally sugars. So firstly, I have 4 foods I've made into a suspension with boiling water. So the first is bread, the second is the cheese spread, the third is a banana, and the fourth is milk. So I'm going to test these 4 foods to see what food groups are contained in each. But so that you can decide yourself which food groups are contained, I'm going to show you the reaction of a positive and negative control, and then the reaction of each test of the 4 groups. So into my test tubes I've already measured. So first one is deionized water. This will form our negative control. It should not react because it shouldn't contain any of the food groups. The second is a positive control. So the first one I'm going to show you is a protein test. So I've got an albumin standard. This is a protein standard, and so it should show us what a positive reaction looks like. Next, I put my 4 foods. So the first one is, so tube number 3 is bread, tube number 4 is the cheese spread, number 5 is the banana, and finally number 6 is the milk. So to carry out the protein test, first thing I'm going to do is to add some sodium hydroxide to each of these tubes. Just add in a bit. I'm just going to do it to all. This doesn't actually cause the reaction, but it makes it possible for our next solution of copper sulfate or biuret reagent to react. So I've added my sodium hydroxide to all of them. I'm now going to take my biuret reagent, which is a lovely blue colour, and again add it to all of the tubes. So when I add it, into the deionized water, it remains slightly lighter blue. If I add it now into our positive control, so I'll add it to, you can see in the positive control it's gone from this nice light blue to a very dark blue, almost a purple in colour. A bit hard to see the purple colour in the video. So I'll now add it, so firstly to the bread. Do you think this contains protein? Does it look more like our negative control or our positive control? Next one, to our cheese spread. Again, does it look more like our negative or our positive control? To the banana, and finally to the milk. For the milk, I'm just going to give it a little bit of a mix. And with all of these, you're going to want to decide, so firstly the bread, is it more like the positive or the negative? For the cheese spread, more like the negative or the positive. For the banana, more like the negative or the positive. And for the milk, more like the negative or the positive. Next food test is for fats. So as before, I've set up the test tubes. In the first is the deionized water for the negative control. In the second is for the positive control. So for fats, I've just used olive oil. Then we have our bread, our cheese spread, our banana, and our milk. So to test for fats, there's 2 different ways of doing this. One, you can put some of the solution onto a bit of brown paper and see if it leaves grease. Another is to add some ethanol to the solution. So I'm going to add some ethanol to each of them. I'll just quickly add to the whole lot and then talk through the reaction. Hopefully that's enough. So to our first one, if I give it a shake, you can see it's kind of one layer. It looks much the same as water. So there's been no reaction. That was our water. To the next one, it's our olive oil. And if I give it a shake, hopefully you can see there's little globules. fat that have formed. Might be quite hard to see in the camera. As I rock it back and forth, there's globules. So this shows that there's fat in the olive oil. Next, if I take the bread, give it a mix, there doesn't seem to be any globules in there. Then for the cheese spread, give it a mix. And if I tilt it like that, you can see some globules. So what does this mean? For our banana, I can see a bit of floating banana, and while on the camera it might look like globules of fat, it doesn't seem to be when I can look at it with my eye. And finally the milk. You can see with the milk there's this massive separation. So, and lots of globules even floating on the top. So what does this mean? So try to work out yourself which ones contain fat. So the next test is the starch test. And as before, in the first one I've got my negative control, which is deionized water. The second tube I've got my positive control. So in this case it actually is just a starch solution. Third is the bread, fourth is the cheese spread, fifth is the banana, and sixth is the milk. So to test for starch, I then add some iodine solution to each of the tubes. So I'm just going to add the iodine solution and then I'll talk you through the result. Okay, so I've added some iodine to all of the tubes. So in the first one, this is our deionized water, our negative control. It's remained much of a muchness in colour. So then our next one, the positive control, instead of that sort of reddy brown, it's gone black, sort of, it's sometimes also said bluish black. So that's the colour we're looking for. So for our bread, do you think it contains starch or not? And mix our cheese spread. Again, do you think it contains starch or not? In our banana, do you think it contains starch or not? And finally, in our milk, do you think it contains starch or not? So the final food test is for reducing sugars. So as before, I put deionized water as my negative control. My positive control is in the second tube, and this time it is a glucose standard. Then I have my bread, cheese spread, banana and milk. So for the reducing sugars, I'm going to add some Benedict solution to all of them. So again, it's blue in colour. It's very similar in colour to the biuret reagent that was used for the protein. So if you look at all of them, they all look pretty much the same blue, but that's because to get the sugars to reduce, I actually have to add it to a hot water bath. So I have numbered all the tubes, but I'm just going to set them all in. Make sure you have labeled them before so you can identify which ones is which. And then you just need to leave it for around 5 minutes, and then I'll show you what they look like once they come out of the water bath. Okay, so now the sugars test has taken place in the water. I can show you the results. So the negative control, our deionized water, has remained the blue of the Benedict solution. The next one is our positive control, so it's gone sort of a yellow-orange in colour. The more sugar there are, the brighter the yellow or the orange will be. So now on to our bread. Do you think this has sugar in it? Or reducing sugars? onto our cheese spread. Do you think this has reducing sugars? Our banana then, do you think it has reducing sugars? And finally, in our milk, do you think there is much sugars in here? So that just shows you how to carry out 4 food tests using simple reagents, and how you can use these to identify the groups of food in various different foods.